Hello everyone and welcome to Instant Biology by Dr. Neela. I am here with you again with a very important topic, that is of DNA vaccines. So many of you would have seen my previous video on DNA vaccines that was in Hindi. So this video would be in English. So for those of you who are more comfortable in Hindi, they can go and watch that video. For those of you who are comfortable in English, can stay with me on this particular video. We would be talking about the principle of DNA vaccines. We would be talking about the mechanism, about how DNA vaccine actually works. We would also be talking about some of the applications related and against what diseases this DNA vaccines nowadays given. So we would be talking, or we would be covering DNA vaccines in a holistic approach. Now, without wasting much time, let us talk about what DNA vaccines are actually. So introducing DNA vaccines are based on plasmid DNA encoding antigenic protein. What do you mean by this? What do you mean by plasmid DNA? So understand this, when I am saying plasmid DNA, plasmid terms comes from the bacteria, or it is the extra chromosomal genetic material. Extra chromosomal genetic material. So it is the extra chromosomal genetic material present inside the bacteria that often gives some additional properties to the bacteria. For example, the bacteria having a particular plasmid, let us say a resistance plasmid, it will offer resistance against a particular antibiotic to the bacteria that is having that particular plasmid. Apart from that, there are some toxins, that there are some plasmids that encode for producing toxins. That means if a plasmid is present inside that bacteria, inside a bacteria, and the function of the plasmid is to produce toxins, obviously the bacteria would be producing toxins, lots and lots of toxins, and because of these toxins, the bacteria around this particular bacteria would be killed. So plasmids basically are extra chromosomal genetic material that are seen in bacteria and offer some advantageous features to that particular bacteria. Now, coming back to our topic, DNA vaccines based on plasmid DNA encoding antigenic proteins. So over here what we are saying is, we are saying that the plasmid DNA, over here the plasmid DNA will be chosen that will be coding for the antigenic proteins. That means if suppose this is a plasmid DNA and this plasmid DNA is coding for, let us say, a protein, and this is an antigenic protein. So when I say antigenic protein, what does it mean? It means that it will be having a foreign proteins property. It might cause some of the diseases inside the host. So these would be the property of the antigenic protein. So what we are doing is, we are choosing a plasmid DNA, and this plasmid DNA would have the property, or this plasmid DNA would be encoding or would be responsible for synthesizing the antigenic proteins. Right? Now these antigenic proteins will again be responsible for causing diseases or causing infections inside a particular host. Right? So you would have understood about the basic concept of DNA vaccine. So what does DNA vaccine involve? So this is what we have discussed up till now. Plasmid DNA injected into the muscle cells of the recipient. Now, if I want to generate resistance against a particular pathogen, what will I do? I will code, or I will take that plasmid DNA that will again be responsible for coding for that particular protein. I will take that plasmid DNA and I will inject it inside the muscle cell of the recipient. Now suppose we are talking about a human cell, or we are talking about a human being, and we want that human being should be resistant against a particular pathogen. Okay, let us say XYZ, any pathogen. So I want to make that human being resistant against that pathogen. So what will I do? I will use a DNA plasmid, and in that DNA plasmid I will make sure that that DNA plasmid should be coding for the antigenic proteins that are present in that particular pathogen, let us say pathogen X. Now when you are taking that DNA, that plasmid, and you are injecting inside the muscles of the human being, what will happen? The human being will start developing response against that particular protein that is being projected. Now again, there is no pathogen over here. There is no pathogen involved over here. Therefore, we are not having an attenuated pathogen. Nothing like that. So we are just having a protein that is being shown up, or that is being identified by our immune system as foreign, and now our immune system will start developing response against that particular protein, not the pathogen. If the pathogen would have been there, then there will be a problem. There would have been a problem because the pathogen might have attacked our cells or might have caused damage, inflicted damage upon our cells. But over here there is no pathogen, only the protein is there, and that too, that protein is being synthesized by a plasmid DNA, and our immune system would be activated by that. So this is what I'm trying to say. Now, why am I insisting upon inserting the DNA or the plasmid DNA inside the muscle? Understand this, that efficient uptake and expression is involved. That means inside the muscle cells, if you are injecting that plasmid DNA inside the muscle cells, then obviously a better expression would be seen and an efficient uptake would be seen. That means the muscle cells would be easily taking up the plasmid DNA, and expression of those proteins on the surface would also be very easy. Next is stability. It is seen through experimental measures that the DNA, that is the plasmid DNA that is inserted inside the the muscle cells, it is seen that the plasmid DNA generally stays stable inside the muscle cells. Next, strong immune response. Muscle cells are very active. Because of this activeness, what happens is the muscle cells produce a small immune response against that particular pathogen. Now, coming to the next thing, depot effect. What do you mean by depot? Now you might have visited a bus stand or a bus depot. What happens is there are multiple buses that are standing over there and waiting for their turn to take the passengers and go on to their route. So what happens is, do all the buses move together? No, the buses move turn by turn. That means the muscle cells over here will serve as a depot, and the release of the proteins or the effect would be shown little by little, just like a depot. From the depot, buses move turn by turn, not together. So that is what I'm trying to say, is the depot effect will be seen. Because of the depot effect, the effect of the DNA vaccine would be prolonged. Okay, so the effect can be seen for a longer period of time because the release of the proteins will be little by little. Understood? Now let us move on to the next thing, safety and accessibility. It is seen that when you are injecting the DNA plasmid inside a muscle cell, the safety feature is always there, and accessibility, that means you can isolate the DNA from the muscle cells. That is also very easy in case of a muscle cell. Now, since we have understood this, let us move forward to the next thing. Host cell takes up DNA. So I am talking about the stages what we will be seeing. So the first stage is what will happen. The host cell, in this case the host cell is the human, let us say. The human cell, the human muscle cell. The human muscle cell is the host cell. It will take up the DNA, and then obviously an immunological response will be created. So what is the immunological response? It can be any response against that particular protein that the plasmid DNA will be synthesizing, right? I will be talking more about this in just a moment. Since antigen is inside the cell, now you know you have injected, this was the cell, let us say this is a muscle cell. This is a muscle cell. Now you have injected your plasmid DNA inside the muscle cells. What will happen? Every cell has the capability to identify the foreign particular protein. Now the same thing will be happening with this muscle cell also. It will be identifying the foreign protein through endogenous pathway. What do you mean by endogenous pathway? In my previous lectures, previous videos, I have already explained that particular pathogen or particular protein can be expressed by two methods. One is endogenous pathway and one is the exogenous pathway. In case of the exogenous pathway, the pathogen or the protein, most likely pathogen, does not enter inside the cell. Okay, so that is the case of exogenous presentation. But if I am talking about endogenous presentation, what happens is the protein or the pathogen comes inside the cell. So what we are doing over here, there is no pathogen as such, but what we are doing is we are directly injecting the DNA inside the cell. That DNA would be synthesizing proteins, and now these proteins are present inside the cells, and because of that, endogenous pathway would be adopted to show the proteins or to exhibit the proteins or to portray the proteins on the surface of the muscle cells. So how it would be done? Endogenous pathway is used up, and in the endogenous pathway we require MHC class 1, right? You would be remembering that. Now MHC class 1 would be used up, and this MHC class 1, let us say that this is a particular cell, this is the MHC class 1. Okay, so this MHC class 1 would be exhibiting some of the proteins, right? And these proteins would be the foreign proteins that have been synthesized by the DNA plasmid DNA. Now, because of this, better activation of the cytotoxic T lymphocytes will take place. You would be knowing that MHC class 1 proteins are responsible for activating the CTL or TC cells. They are responsible for activating TC cells. So now what will happen, the TC cell will come in. Let me just draw it like this. Let us say this is a TC cell. It will come over here, and with the help of TC receptor, it will identify this particular pathogen or this particular protein, not pathogen, this is a protein. This protein is having a foreign origin, and now what will happen? An immune response will be mounted against no pathogen, but only against the protein. But what it will do, it will equip your immune system to tackle the condition in a better way, so that and be ready if in any case the actual pathogen comes at a later stage, your immune system is in a better position to tackle it, because it has learned that what are the proteins that will be involved in that kind of a pathogen and how we can tackle it. So this is the concept. Coming to the next thing, inside the cell the antigen can remain in two forms. Understand this, that either the DNA that you have inserted inside the cell, inside the muscle cell, it can remain there in two forms. What are the two forms? One form will be that it can directly combine with the chromosomal DNA. One way, it will directly combine with the chromosomal DNA, and the other method that it involves is that it can integrate into the host DNA. This is the first method. Or it can remain as a separate entity inside the cell. That condition also can persist, right? So first condition is it can stay in the episomal form, that means separate form, and the other condition is that it can be integrated inside the original chromosomal material. Now, so this you have understood, I believe. Now let us move on to the next slide. Coming to the aim. So what is the aim in DNA vaccines? What do we aim? So the aim is that DNA plasmid uptake by antigen presenting cells. What I want is that the antigen presenting cells should somehow take up the DNA, right? So what are the antigen presenting cells? The antigen presenting cells can be, for example, let us say B cells, for example dendritic cells, for example macrophages, right? So these are the antigen presenting cells. So what is my aim is? My aim is that antigen presenting cells somehow uptake the DNA plasmid, right? If the antigen presenting cell, so that method would directly happen. That means the method that I just talked about, that TC cells will be activated and immune response will be generated, that is fine. But if somehow the DNA plasmid is uptaken by the antigen presenting cells, what will happen? Antigen presenting cells, play a major role in antigenic responses against these vaccines. Right? So understand this, that whatever vaccines you are giving, if the antigen presenting cells get involved, what will happen? Obviously B cells, if they will get involved, humoral immunity will also get involved, right? You would be knowing that the B cells have written antigen presenting cells also involves the B cells. So if the B cells will be activated over here because of the we are talking about the dendritic cells, B cells and macrophages, if they get involved, what will happen? The humoral immunity will get activated. T cell related immunity, we have already talked about how it will get activated, but over here the humoral immunity will also get activated if the antigen presenting cells take up that DNA, right? Now, provide protection against influenza, rabies, etc. Now, these DNA vaccines have been majorly used up against influenza, against rabies. Booster doses have also seemed to be required. Now, you should be giving booster doses in case of the DNA, and the booster doses, what it should contain? It should be either containing the protein antigen itself. Remember, we had started with the protein antigen, that the same antigen proteins that were made by the plasmid DNA, you can directly give the protein antigen itself, or you can also give the DNA sequence in the form of a vector. So we had given the DNA sequence in the form of the vector previously in the form of plasmid, right? So that same can be done, or you can directly inject the protein itself. That can also be done in the form of a booster, but booster doses are required over here. Now, let us move forward and talk about the next thing. Many times, CpG DNA motif is added to some vaccines. So what do you mean by CpG? Let us understand this. So C means cytosine, P means phosphate, G means guanine. So CpG islands, this is called as CpG islands. So what I'm trying to say is, suppose this is the DNA that you had injected. Now to the DNA, what you have to do is you have to insert a CpG, 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 something like this. Basically, what is this? This is a CpG island, and this CpG island is responsible for the increasing the effect of the vaccine. So when you are adding a CpG DNA motif, it will obviously extend, or it will obviously enhance the effect of the vaccine. Now, what are the advantages of a DNA vaccine? Encoded protein expressed in the natural form. Now you are not creating any kind of a manipulation inside the DNA. What you are doing is you are directly using up that DNA that will be responsible for coding those proteins. So the proteins that will be synthesized over here would be in completely natural form. What do you mean by natural form? In the form that they are present naturally, in the form that they are produced by the bacteria or pathogen naturally. That would be directly produced over here, and because they are produced over here, the response would be very quick, the response would be very efficient, right? So encoded proteins expressed in the natural form. So no denaturation or modification is involved over here. We have created no modification or no denaturation of the DNA. Now, strong stimulation of both arms of the adaptive immunity. I have told you that both the arms, that means the T cell arm, those are cell mediated immunity. So let me just write down over here. So over here, cell mediated immunity is also activated, and the B cell, or let us talk about the humoral immunity. Humoral immunity is also activated. So cell mediated and humoral, both are activated. Now, since both of these arms are activated, now we can say that immunological memory is also involved. You would be knowing that when the adaptive immunity gets activated, obviously the immune or the memory response also takes place. That means your body will be remembering that how it tackled a particular pathogen and create the responses later on. If the pathogen comes later on, then the same responses will be created. Next, no refrigeration required. Now over here you have seen that DNA is only used in DNA vaccine. What you are doing? You are injecting the plasmid DNA only. So DNA does not require any refrigeration. You can directly keep the DNA at the room temperature. So the vaccine does not require any special treatment. You can keep it in the normal or the room temperature. So that is not the case in most of the vaccines. You will have to keep them in refrigerated conditions. Coming to the next point, vector can be custom tailored again and again. Now this is a very, very important feature of the DNA vaccine. That means if you have made the vector once, right? So what I'm saying is, if you have made the vector once, suppose I have made the plasmid once. Now what I can do is I can keep on injecting the proteins from different types of pathogens. So the vector is same, the plasmid is same, and I am again and again changing the DNA, the important DNA or the DNA coding for the protein, pathogenic protein. I am changing that only again. So let us say that this was for pathogen number 1. I can convert it for, let us say this is for pathogen number 2, pathogen number 3, so on. What I can do is I can just remove this and I can insert this, and I can do this again and again and again and again. Right. So West Nile virus, let me give you an example of a DNA vaccine. So a DNA vaccine against West Nile virus was given to the horses, and a very prominent effect was seen in case of the DNA, in case of the horses against this West Nile virus. Nowadays, currently a lot of work is being done on cancer. So most probably, most of the books and most of the authors, they predict that a good vaccine, we could be seeing, or we could be observing a good vaccine that would be DNA vaccine in nature against cancer in a very short duration of time, and that is not very far away from now, right? So this was all about the DNA vaccine. I've told you on which organisms, basically against rabies, against influenza, this DNA vaccines are used. Apart from that, I have also given you an idea that what is the principle and what is the mechanism of DNA vaccines, the applications of DNA vaccines. I've given you everything. Now I have some very important announcement that I want to make to all of you. You might be knowing that GATE biotechnology batches have already started. So a recent batch we had started on 23rd. Let me just write it down over here. So we started a recent batch. Just give me a minute. Yes. So we started a recent batch on the 23rd July. The timing of the batch is from 7 a.m. in the morning to 8:30 a.m. Also, an evening batch is also running. So this was about the morning batch. An evening batch is also running. That happens from 6 p.m. in the evening to 7:30 p.m. So these are the two batches that are currently running for GATE biotechnology classes, and in these batches you get everything. By everything I mean literally everything. That means you get daily practice problems, you would be getting the recordings, you would be getting the notes, you would be getting the mock test, you would be getting the tests after every chapter, and the most important thing that you would be getting is the revision. So we would be completing the course in around January first week, and the rest of the time that we have at our hands would be done or would be given to revision. So this is the important information that I wanted to give about the GATE biotechnology examination, GATE biotechnology sessions, sorry. And I have one more important information. I've already told you this previously also, that we have recently launched an umbrella course on UDAN. So what is UDAN? UDAN has been launched in order to help the students who wish to apply to foreign universities, either for master's courses or for PhD courses. So in UDAN, what do we do is we provide, the basic program is that we provide general consultation to students, and we directly create a meeting with the expert to the student. They can directly interact. I would also be there in order to give my inputs, so they can directly interact, and the students can directly ask their queries to the experts, and they can get their queries sorted. So that particular general consultation is as of now at very affordable price. The price is just rupees 299 for 45 minutes. Okay, so you can ask your queries to the expert for 45 minutes, and the price is just rupees 299. Now if you find that particular program interesting, you can contact us again and ask for the help in SOP or help in your letter of motivation or things like that. So that is another services that we offer, but that is for extra charge. But this, again, you can ask your queries to the expert, just 45 minutes for just rupees 299. So both of these things I wanted to announce. The details of both of these things you can get from us. You can contact us on 8299134528. So this is a WhatsApp number. You can get in touch with us on this number, and we would be happy to provide you more information on both of these courses. Thank you so much and have a good day.